All right, welcome to the fuel sources for muscle and exercise metabolism discussion. This is a pretty long lecture, so I'm splitting it into two parts. This is part one. In this nutrition related class, we do have to get a better understanding of what happens to the fuel that we consume, how it's stored, and ultimately how it's metabolized to provide us with ATP or energy to drive our muscles to do meaningful exercise. So that's what this discussion is all about. Um, we'll talk about the macronutrients, we'll talk about different metabolic pathways, we'll discuss some enzymes. The important ones mainly, there's a lot of enzymes involved. We won't get into every single one, nor will we get into every single reaction because there's far too many to discuss, especially in a class like this. So yeah, we'll talk glycolysis, Krebs cycle, a little bit of phosphagen system as well. And then we'll sort of finish things up talking about the regulation of metabolism, because once you get all the pieces in place, that's great, but we need to understand when they get turned on, when they get turned off, and how they're used under different circumstances. That's really kind of the application of all this. So understanding the mechanics first, then the application. All right, so metabolism and bioenergetics. Yes, as you already know, surely, nutrients of the foods um, are the substrates that we use for metabolism. Come through carbohydrates, fats, and proteins. You have a little bit of background on the three so far in this course. Uh, Obviously, we obtain these through food sources. Um, our bodies and each cell basically in our body contains different chemical pathways to take those substrates, right? We're going to start calling them substrates, to take the carbohydrates, the fats, and the proteins and convert them into usable energy. Our body has preference for carbohydrates over the other two, but the other two are actually quite important and sometimes very important. So really, all three are going to be discussed in this lecture. Um, the form of energy we derive from food ultimately is in the form of adenosine triphosphate or ATP. And ATP serves as the immediate fuel source for energy for most body functions, and especially in the case of a class like this, we're talking about muscle contractions. We're all about muscle contractions. It's all about ATP. So being able to regenerate it, to produce it, to get it from the reactions from breaking down our food is what this is all about. Um, so let's introduce metabolism. Metabolism, by definition, it's the total of all chemical reactions that occur in the body. There's two primary types of metabolic reactions. There's anabolic reactions, which is the synthesis of molecules, so taking smaller molecules and making bigger ones. And then there's catabolic reactions, which is taking larger molecules and breaking them down into smaller ones. Uh, Bioenergetics is a term that describes the converting of foodstuffs, fats, proteins, carbohydrates, like we mentioned previously, into energy. So metabolism. Metabolism ultimately, even though it is, yeah, the sum of all chemical reactions in the body, it's really all about energy transfer. We're transferring energy from perhaps an apple that you ate from lunch and transferring the energy from that apple to help fuel your muscles to produce meaningful muscle contractions to maybe help you run. There's different ways to transfer energy and to liberate this energy. There's aerobic and anaerobic pathways. We'll talk about each of those. Um, and ultimately, the fuel source and from the substrates, it isn't necessarily, the whole category of carbohydrates. It's carbohydrates in their simplest form, which is going to be glucose. Fats are going to be converted from free fatty acids and proteins as amino acids. So it's really not the whole substrate in its whole form. It's after that whole digestion and absorption process, and then ultimately storage process after absorption, when these substrates are available to be broken down. So didn't want to go that fast. So the body stores of fuel and energy. So this is just this slide and actually the next one as well. Just to give you sort of big picture idea, how well does a body store energy? How much energy do we have, right? Oftentimes the body is compared to like an automobile and sort of like, hey, if you can fit 10 gallons of gas in your car and your car gets 20 miles per gallon, you kind of know how far you can go before you run out of gas, right? Well, you can kind of do the same thing with humans. And when we look at our two primary fuel sources, it is carbohydrates and fats. The body will use protein. It doesn't like to use protein. It likes to keep protein. But if we look at carbohydrates and fats, we can get an idea of how much energy we have stored in each of them. So when we look at this table, we can at least go over to the kilocalorie section and go, yeah, storage form or storage of carbohydrates. We find a lot in the liver. We find probably most of it or most of it in our muscles, and there is some floating around in our blood. Ultimately, we have approximately, this is, as you see at the note on the bottom, right, average size human. Um, Average size individual has about 2,500 calories worth of carbohydrates stored in their body. Our body will not usually let us exercise to the point where those are all gone, but nevertheless, even just 2,500 calories isn't that much. If you think of meaningful exercise, right, exercise of a moderate to high intensity, you could easily eclipse the 10 to 15 calorie a minute zone in terms of burning carbohydrates. And so we're looking at a pretty limited supply of carbohydrates as a fuel source if you're going at a pretty good clip. Fats, on the other hand, we have a pretty good supply of fats. Um, we store fats throughout the body. Our subcutaneous and visceral fat stores um, are available to be used as a fuel source. We also store actually quite a bit of fat within our muscles, and our muscles prefer, and that's why I just circled this 161 grams. When exercising, our muscles prefer fat from within the muscle. It's just closer, right? It takes a little less effort to liberate fat in the muscle as opposed to liberating fat from the abdominals, to drop that into the blood, to deliver it in the blood to the muscle, absorb it into the muscles, and then utilize it for a fat source, fuel source. Too much energy, right? So intramuscular fat is actually the predominant fuel source during exercise for the most part. Up to 70 to 80% of fat burn comes from within the muscles. Uh, Nevertheless, when we compare total fat storage, and again, this is a 12% body fat individual who weighs 143 pounds, so not that much fat and not that big of a person, um, somewhere in the neighborhood of 75,000 calories, um, compared to the 2,500 calories uh, of carbohydrates. We're looking about, what is that, 30 times as much, somewhere in that zone. So a lot more fat storage than carbohydrate storage in our bodies. This table is a little bit more detailed. It actually uses the same average individual, which is convenient, although this person has a little bit more body fat. But it does kind of the same thing, just with a bit more on the details. So, and maybe that main detail is how long we can exercise um, using that fuel source, right? And so about 20 minutes worth of liver glycogen, about 80 minutes worth of muscle glycogen. For those of you who do high intensity exercise, especially high intensity or moderate high intensity endurance type of exercise, some of you may have hit the wall before and gotten to that point where you're just tapped, right? Cyclists call it bonking. I think runners call it the wall. I'm sure there's other terms for it as well. When does that usually occur? Yeah, usually around 90 minutes or so. It depends. Depends on you, your fitness level, what you're eating or drinking during exercise. But a, a lot of that is attributed to, not everything, but a lot of that is attributed to pretty much tapping your carbohydrate stores. Once you kind of run out of carbohydrates, what are you left with as a meaningful fuel source? Yeah, your fat and your protein. Can we liberate fat and protein fast enough to get ATP to sustain high intensity exercise? We haven't answered that question yet, but we will, and that's part of what this talk is about. So the answer is pretty much no, and so that's why hitting the wall or bonking is so difficult to overcome without the carbohydrate feeding, and really it takes some time to recover from that. And like I said, there's other factors involved there, not just potentially running out of carbs. Yeah, looking at fat as a fuel source, you got about 5,000 minutes. Protein, we can go a lot longer off our protein stores than we can carbohydrate stores. We don't want to do that because ultimately that protein is coming from our muscle mass. We want to save our muscles. Um, anyway, so some good big picture types of ideas about our body's ability to perform and utilize the food that we consume after storing it. Again, kind of giving you that idea of the gas tank in a car kind of thing. It's like, yeah, what kind of gas tank do we have as humans? Uh, Turning the page, let's talk about enzymes for a minute. Enzymes are a huge part of our existence. We really don't appreciate them nearly enough and often take them for granted. We've talked about enzymes in digestion quite a bit. and their role in breaking down larger food components into their basic components, right? Breaking them down, breaking bonds. Um, Generally speaking, yeah, enzymes are biological catalysts. They speed up chemical reactions without being involved in the reaction or altering the free energy release. So enzymes are fantastic. They help chemical reactions occur more quickly and more thoroughly without messing up the reaction. Uh, Metabolism is all about the function of enzymes and the presence of enzymes. And maybe more importantly, this other bullet down here, um, metabolism is all about turning enzymes on and off. And we have what are called rate limiting enzymes. And and they're also called allosteric enzymes. So the more technical term is allosteric. But rate limiting enzymes, and I put that little graphic of the stoplight there, these are enzymes that truly control metabolism. If a rate limiting enzyme, let's say in glycolysis, it's the enzyme PFK or phosphofructokinase, if it's turned off, glycolysis, which is a breakdown of glucose for energy, will not occur. If it's turned on, we're going to be burning plenty of glucose as a fuel source. So these enzymes, these allosteric enzymes, are the ones that really control metabolism. And thankfully, the way they're designed is, yes, they have triggers that turn them on and that turn them off. And the consequences, because always a good discussion question about these enzymes is, why do we need to have enzymes be able to be turned on and off? Well, think about, if we just go straight back to the PFK, right, the phosphofructokinase. Let's say, for example, it was an allosteric, and any time that the presence of, and it's glucose 6-phosphate is what it reacts with. Um, Anytime that showed up, then if PFK didn't have the ability to get turned off, glycolysis would occur, which means even at rest, right, glycolysis would be going on at an uncontrollable rate. We would not be able to eat enough to keep up with our burning of energy. We would burn carbohydrates as soon as we consume them. So the fact that we have enzymes that can be turned on and off, it stops metabolism. It slows metabolism down when we don't need it. When we're at rest, hopefully most of you are at rest watching this, you don't need to have a high metabolism, right? So PFK should be turned off for the most part, unless you're watching this while you're on the treadmill or something, which is possible. Okay, so let's talk about these energy systems. I mentioned anaerobic before, and the term anaerobic just means without oxygen being present. So we have two energy systems that can function fully without oxygen being present. In the case of metabolism, exercise metabolism, we're talking about the muscle cell. Without oxygen being present in the muscle cell, we have two forms of metabolism, so two ways to generate ATP that can function. This is really important because we don't always have ample oxygen in the muscles. Um, that's a that's a whole topic for another class, how it gets there and why it's not there. But without oxygen, right, we still can produce or utilize, create energy to use in a meaningful way. And so the first anaerobic system we'll mention is called the phosphagen system. Some people call it the ATP-PC system. Some people call it the creatine phosphate system. There's a lot of sort of terms that describe the same system. The phosphagen system is our most immediate source of ATP regeneration. We'll talk about the details of that in a minute. The other anaerobic form of metabolism is glycolysis, and we just mentioned that a minute ago. Glycolysis is the breakdown of glucose or glycogen. Glycogen is a storage form of glucose um, for energy. And so it has two different phases. Excuse me. We'll talk about those phases. We'll talk about where the products of those phases go to help produce more ATP in other parts of metabolism. We'll talk about all that stuff. Just know from this slide, yeah, phosphagen and glycolysis, those are our two anaerobic energy or metabolic pathways. Aerobic, on the other hand, is a type of metabolism where oxygen must be present. And keep in mind, all of this is going on in our muscle fibers, in our muscle cells. So at least to orient yourself, where are we talking about? What is this stuff going? Where is it going on? We're in the muscle cells, aerobic metabolism. There has to be oxygen molecules floating around in there, usually in the mitochondria. The first pathway we'll talk about is a Krebs cycle. It also has multiple names, a citric acid cycle or the tricarboxylic acid cycle. Krebs cycle seems to be the most popular, at least. And then we also have something called the electron transport chain. These two systems work in conjunction with each other. They're often called oxidative phosphorylation as a group, although it is quite separate. The oxidative phosphorylation is more of the electron transport chain, but often people will say that, or sometimes people use the term oxidative metabolism. We'll talk about both of those in detail. So the information on this slide is just sort of introductory type of stuff. All right. So let's introduce our first metabolic pathway, and we're going to go with our two anaerobic ones first. So the first one is the phosphagen system. I will try and call it the phosphagen system as much as possible without being persuaded or distracted into using the other names. The phosphagen system is our most rapid means of regenerating ATP. The phosphagen system has two total reactions, but really only one reaction. The first reaction of the phosphagen system is its primary reaction, and it is only a one reaction pathway for us to regenerate ATP. It's pretty incredible. So the regeneration of ATP from the phosphagen system is on this top line right here. And so what we have, and this is going to take a little bit of your physiology, metabolic background to at least get oriented here, but a molecule of ADP, and ADP is adenosine diphosphate. So it's an adenosine molecule with only two phosphates on it. Our body likes adenosine to have three on it, right? ATP to be used as an energy source. And so really, and sometimes in the most simplistic way possible, metabolism's sole purpose, as described by others, not myself, its sole purpose is to reattach phosphates onto ADP. That really drives metabolism. So when ADP is present in our muscle cell, metabolism turns on. Hey, wait a sec, there's an ADP there. We need to put another phosphate on it to make ATP. That's all our body wants to do. So when we start to use our muscles, we use ATP to move them, and the splitting of that ATP, right, the release of energy from that ATP gives us an ADP, adenosine diphosphate, and that little phosphate molecule that got split off of the ATP is now floating around as well. So I should have put this in graphical form. Hopefully this is review for all you anyway, but just being mindful, I'm trying to at least describe where these substrates are coming from. So this ADP came from the splitting of ATP. The phosphagen system is really good at adding that phosphate back onto the ADP. And the way it does it is it finds that ADP molecule and it adds, it combines it with a molecule of what's called creatine phosphate. So the CRP is creatine phosphate. And creatine phosphate is a molecule of creatine, and it's an amino acid, and a phosphate molecule. So that phosphate that got split off of the ATP when it released its energy, somewhere along the line, we'll talk about that in a minute, it found a creatine molecule. And so we have this molecule of creatine and phosphate combined. They get together with the ADP, and the magic of an enzyme, this enzyme is called creatine kinase, CK. Creatine kinase is the main enzyme of this reaction. It essentially tears this phosphate off the creatine, adds it to the ADP, and it gives us a molecule of ATP, which is all we want, and then that poor creatine is now all by itself. And that creatine, thankfully, can now join up with another free inorganic phosphate that's floating around to reform creatine phosphate. A lot of you may have heard of creatine use as a supplement, and the purpose of individuals using creatine is to increase concentrations of creatine phosphate. Creatine doesn't come in the form of creatine phosphate, it comes in the form of creatine when we supplement it, but by increasing this concentration is more likely to give us greater creatine phosphate when we need it, and it is quite effective. So hopefully that was a thorough enough job of that first reaction of the phosphagen system. That's it. ADP plus creatine phosphate, mix it with that magic enzyme creatine kinase, CK, and we get our ATP. And it can happen over and over and over again until, and hopefully some of you are asking, yeah, what happens when there's too much ATP? Let's say it's really high intensity exercise, you're going for a sprint, and creatine phosphate isn't keeping up. What happens? Well, that's the second reaction of the phosphagen system. So the second reaction kicks into gear when there's a situation of usually extremely high intensity exercise where the phosphagen system can't keep up with the production of ADP to convert it to ATP. So we take two ADP molecules, and if you think of it visually, right, we have two adenosine diphosphates. So we have four total phosphates hanging out. All we really need is three on an adenosine molecule, and we got our ATP. And so this second reaction, which is catalyzed by the AK molecule, adenylate kinase, this reaction takes two ADPs, pretty much strips one of the phosphates off of one of the ADPs, adds it to the other, and so we end up with an ATP, and now a new molecule of something called AMP, adenosine monophosphate. So we still have four phosphates, just three on one adenosine and now one on the other. This ATP can be immediately used for energy release. Unfortunately, this AMP can't do much with it. It is, however, as you'll see much later in this talk, a very powerful stimulator of metabolism. When AMP starts to show up in our muscles, it turns everything on. It lets the metabolic pathways downstream, so to speak, lets them know that, hey, they're going to have to start making some ATP because the phosphagen system is starting to fail. So just some side notes on this. Yes, and adenosine monophosphate, like we just talked about, is the activator of the allosteric enzyme phosphorylase. So it turns on phosphorylase, which turns on glycogenolysis, breakdown of glycogen as a fuel source, and phosphofructokinase, PFK, which we already mentioned. AMP turns both of those on. So they pretty much, AMP is a stimulator of the next phase of metabolism that we'll talk about, which is glycolysis. Yes, the ATP production capacity of the creatine kinase reaction, this first one up here, relies on a store of creatine phosphate. Yep, we mentioned that. Stores of creatine phosphate can be depleted in 10 seconds or less. Some of you, maybe in an exercise physiology class, have done what's called a Wingate test. And during those Wingate tests, it's often pretty obvious to see a phase of your peak energy production, so your peak energy output, kind of one and the same during that test, right? And all of a sudden there's a pretty dramatic drop off, and then you sort of tail off consistently after that. That first peak of a Wingate test is all about the phosphagen system. That first phase usually lasts, and most people, varies, about 5 seconds. And that is a real good indication that, yeah, the phosphagen system just got tapped, and now we're relying on glycolysis. So once depleted, replenishment of creatine phosphate can't fully begin until recovery has started. So, and that is passive recovery, really, is optimal. So it's really hard to recover creatine phosphate unless we stop. That does have implications for those of you who do weight training and sprint training out there. Is it best to keep moving as you recover? To help clear out things like lactate, yeah, and to help you recover, maybe not vomit, sure, but to recover creatine phosphate, don't do anything. So many things. So here's a cool graph, and it pins up or posts up ATP and phosphocreatine during a sprint. So this is a entire 14-second sprint, so not even a full standard Wingate, which is usually 30 seconds, right? In blue we have ATP, in red we have phosphocreatine, percent of resting value. And so all this is showing is how much at the beginning of this sprint we have 100% of resting value of ATP and phosphocreatine. By the end of the sprint, what's happened? And what's interesting, and at least what is important to note, and the question is asked down here, why does the body use phosphocreatine first? It's a good question, right? Because if you look, right, the first 2 seconds here from our storage capacity of phosphocreatine, we go from about 100% storage to about under 50. So we use up half of our phosphocreatine the first 2 seconds of a sprint. However, what happens with ATP? It stays pretty stable. I mean, we drop maybe down to 90%, it hovers right that 85-90% zone up until about 10 seconds. Our body prefers to keep a store of ATP intact. It's emergency energy. It signifies homeostasis. We're always aiming to maintain homeostasis as long as possible. ATP is more powerful in establishing homeostasis than keeping a store of phosphocreatine. Phosphocreatine is the fuel source. ATP is the stored energy that we're really trying to make sure we have in case there's an emergency. So our body's doing its best, right? The first 10 seconds, yeah, we're holding on to that ATP. Phosphocreatine's really doing its job. And then by the end of this 14-second sprint, this person's probably getting pretty exhausted. Even ATP stores start to drop. And actually, it is indicated here that, yes, exhaustion was reached after 14 seconds. Pretty short sprint. Pretty short sprint. Um, so yeah, that's pretty standard procedure in our bodies there. All right, let's move past the phosphagen system. We haven't talked about a food source, and actually, phosphagen, I didn't mention it, right? Are we using, what is our fuel source? What is it? Was it from your bagel that you ate? Was it a carbohydrate? Was it a fat? Was it a protein? If we're saying that creatine phosphate is the fuel of the phosphagen system, it could be argued that protein is what fuels the phosphagen system. It's not generally considered a metabolic pathway that uses sort of the macronutrients, but you have to argue, yeah, creatine is made up of amino acids. So, all right, let's talk about carbohydrate metabolism. The way carbohydrate metabolism is going to be presented for this class is in 4 stages for the complete breakdown of carbohydrates. So the complete breakdown will involve some aerobic and anaerobic pathways. So just be mindful of that. We're going to sort of use a 4-stage approach. Carbohydrates, I'm going to abbreviate them CHO pretty much from now on, are what are considered a preferred fuel source by our body. And that's an interesting way to talk about them. But when carbohydrates are present in our body will use them, our muscles will use them preferably over fats and proteins. Carbohydrates are easy to access, easy to break down. Not to say that fats aren't easy to break down, but when the two are there together, oh yeah, let's just take the carbs. It's easy stuff for the body, or easier at least. So anaerobic ATP production, which is the process of glycolysis, is what we're going to talk about first. The complete breakdown of a carbohydrate does require oxygen. So glycolysis doesn't completely break down glucose or glycogen molecule, as you'll see in a minute. So glucose is the molecule that we're going to talk about being broken down, even if it starts as a glycogen molecule. You'll see that in a second. We have some graphics that show that. So excess glucose in our body is stored as glycogen. We see that in the muscles and the liver. Really, glucose is only found freely in our blood for the most part. The term glycogenolysis won't be used often. However, it does occur often. Glycogenolysis describes the process of the breakdown of glycogen to glucose for metabolism. So really, when we're talking about muscle metabolism, a majority of the breakdown of sugars, of carbohydrates, is glycogenolysis and not necessarily glycolysis, because that glucose originally started as a glycogen molecule. All right, so an old school graphic, but I still use it because it's just simple, right? So we have our 3 primary macronutrients across the top here, fats, carbs, and proteins. I obviously want you to focus on the middle there, where that's sort of surrounded by the red dotted line. We're going to talk about carbohydrate metabolism first. We will get to proteins and fats later. We're going to start with glucose, go through the process of glycolysis. That'll be phase 1. Phase 2 is a process of going from pyruvic acid to acetyl-CoA. That's going to be stage 2. The Krebs cycle, stage 3, electron transport and oxidative phosphorylation, stage 4. So our 4-stage breakdown of carbohydrates is captured in this old school graphic. And again, we'll keep coming back to it just to get oriented as to where we are in the big picture of metabolism. So there's our 4 stages again. There's their names at least, and those names might be useful, especially when it comes to discussing this at a later time. Glycolysis, pyruvate to acetyl-CoA, Krebs cycle, and the electron transport chain. There are so many graphics out there that help demonstrate the breakdown and the process of glycolysis all the way through the electron transport chain. You're all surely more internet savvy than I am and are able to use resources at your disposal. There's wonderful YouTube clips and TED Talks and other animated versions that take you through the process of carbohydrate metabolism. Take advantage of as many of those as you see necessary to get a good understanding of the process, because it is really much more complex than I present. And I won't assess you at a level that's much more complex than I present. However, the better you understand it, the better you can apply it. And really, the understanding of metabolism to sport and even sport nutrition, of course, is really essential. So let's talk about the purpose of glycolysis. The purpose of glycolysis, generally we get some ATP from it, first and foremost. We barely get any. We get 2 ATP from one glucose molecule going through glycolysis. It's not very much. 2 ATP total. We get a couple of byproducts. One of the byproducts is 2 molecules of NADH with hydrogens, and we'll talk about NADH later. Those actually give us some more ATP just later on in metabolism. And then glycolysis provides us with other substrates. We end up with pyruvate, potentially lactate, and these substrates can travel, be transported, shuttled, taxied, whatever, to other pathways and used for metabolism. We'll talk about that as well. So there's a problem with glycolysis, and it isn't such a problem, but it is still discussed as a problem. I think it's a problem. The process of glycolysis liberates some hydrogen ions. And this is before even we have the production of lactate or lactic acid. And these hydrogen ions contribute to the environment in the muscle. And as you probably already know, free hydrogen ions contribute to the pH of an environment. And the more free hydrogen there is, the lower the pH or the greater the acidity. When we talk about exercise performance and fatigue, one of the primary causes of fatigue is a change in the environment of the muscle, and that change in environment, increasing acidity, can affect metabolism quite dramatically. And there's pretty good evidence out there that, well, it's not even evidence, it's just fact that if you throw enzymes into a acidic medium that they're not able to function in, they won't. And therefore, enzymes that are part of metabolism, when the acidity becomes too great for them to function, they'll stop, and therefore metabolism stops. And so that problem of glycolysis is that it's a little bit self-limiting because of the acidosis that occurs when we rely on glycolysis too much. That in turn slows metabolism down. So the longer we try and rely on glycolysis, the quicker we're going to probably fatigue. All right, so glycolysis. Let's orient ourselves again. We're in the muscle fiber. Inside the muscle fiber, we have carbohydrates, we have fats, we have plenty of protein in the muscle fibers themselves. In the muscle fibers, we have mitochondria, we have all the great sort of cell organelles that you'd expect in a human body cell. We have blood flow going around, there's plenty of fluid, all that good stuff, right? So we're at the point where we want to start regenerating ATP, and our muscles are looking for something to get that fuel from. What's hanging out there? Well, we got glycogen. And so glycogen stored in the muscles, our body knows that it has energy in it. It's not a mystery. And so when we initiate movement, or want to initiate the process of the breakdown of glycogen, the enzyme phosphorylase literally cleaves off individual glucose molecules from the glycogen. And I don't have a graphic of it, but glycogen is and it's not really snowflake looking. It's pretty much a bunch of chains of glucoses all sort of bound together with multiple ends. So glycogen has all these strings of glucoses that are joined, and there's probably hundreds, maybe thousands of different ends. Phosphorylase just finds one glucose hanging off there and just cuts it off. So we have a molecule of glucose now free. Phosphorylase, when it does that, converts or enables a phosphate to be added to that glycogen that it cut off, or sorry, the glucose that it cut off of glycogen. And so we end up with a molecule of glucose 6-phosphate. So just to recap, glycogen hanging out in the muscle, everything's good, you're just sitting there. Let's say you want to take a break and you pause this video right now and you want to sprint over to the fridge to grab a glass of juice. As you're sprinting, you need ATP to do it. Phosphorylase will cut off of glucose from that glycogen, add the phosphate to it onto the 6th carbon, just to be specific, and you end up with a glucose 6-phosphate molecule. So it's a glucose molecule with a phosphate hanging off of that 6th carbon. Yes. Or, still in the muscle, right? We could take a glucose from the blood, because that's probably where we're going to find free glucose, And if we take glucose from the blood and introduce it into the muscle, here you go, thanks to the GLUT4 transporters, all that wonderful stuff going on with insulin. Glucose, with the help of the enzyme hexokinase, can add that phosphate to itself to give us that glucose 6-phosphate. So there's two different ways to get to this whole glucose 6-phosphate, and this is essentially the second substrate of glycolysis. So glycolysis is the breakdown of glucose, glycogenolysis, breakdown of glycogen. I'm kind of combining here, right? So the breakdown of glucose into glucose 6-phosphate requires the enzyme hexokinase, and some of you probably picked up on this already, it also requires the breakdown of ATP. So it actually takes energy to start glycolysis. ATP releases its energy and it actually enables that phosphate to get added to the glucose. So either way, we started with glycogen, we started with glucose. Ultimately, after the first reaction, we end up with a glucose 6-phosphate. Glycolysis has begun. So from glucose 6-phosphate, we go to fructose 6-phosphate. Don't worry now about the substrate so much. What becomes important at this fructose 6-phosphate stage, however, is this enzyme right here. And you've noticed, we've talked about this one before. This is a phosphofructokinase reaction. And so when we get to actually fructose 6-phosphate, the phosphofructokinase reaction occurs. And this is a allosteric enzyme, as you know, and PFK, I'm going to start calling it from now on. PFK, it can be stimulated, it can be turned on, or it can be turned off. And so if it's off, fructose 6-phosphate and even glucose 6-phosphate will start to build up a little bit. They can actually go back in reverse and get converted back to glucose if phosphofructokinase never turns on. Then we're not needed anymore. We can go back to being glucose through the process of, they call it gluconeogenesis, even though it pretty much was still glucose. But in the process of gluconeogenesis, we regenerate glucose from the partial alteration of the glucose molecule. But if all things are good and we're ready to exercise and we're starting the process, the phosphofructokinase will be turned on. We got a green light here, and we need a little bit more ATP to kick this off. We're going to add another phosphate to the fructose 6-phosphate molecule to end up with a fructose 1,6 bisphosphate. In other words, we have fructose now with a phosphate on the first and 6 carbons of that molecule. Not to worry too much. I'm not going to pick on you on those details. But the ATP had to come in to help with that. So up until this stage, let's say we're at the fructose 1,6 bisphosphate stage, we've spent 2 ATP, and we've had to be sure that PFK was turned on. So far, glycolysis is pretty much a bust. Things get better. So after the next reaction, we end up with this glyceraldehyde 3-phosphate and dihydroxyacetone phosphate molecule. It gets split, and it getting split is a big deal. So we still have our 6 carbon intact in this phase. What ends up happening is this triose phosphate isomerase splits the molecule in half, and then we end up with 3 identical molecules of glyceraldehyde 3-phosphate. So we have 2 identical molecules now. They each have 1 phosphate on them. Everything is looking good for what is called phase 2 of glycolysis. So what's on the screen right now? Phase 1 of glycolysis. It cost us 2 ATPs. Phosphofructokinase had to be turned on. We may have started with glycogen. We may have started with glucose. Depends. So let's talk about phase 2. All right. So phase 2. Phase 2 is called the energy generation phase. It gives you a hint. We finally get some energy back. Everything happens in duplicate now. Because we have 2, 3 carbon molecules, they go through the same process, but everything, right, because there's 2 molecules now, everything happens twice, essentially. So early on in phase 2, we see the formation of a molecule of NADH. So we have this molecule of NADH, nicotine adenine dinucleotide. We'll talk about it a little bit more later. So all we need to know for right now is that we end up forming 2 molecules of NAD, and then nicotine adenine dinucleotide can shuttle, it can carry hydrogen ions. That's a big deal. So we form 2 molecules of NADH. Awesome. The 7th step of glycolysis, we actually take an ADP and we add a phosphate to it and we get an ATP. And because that's happening twice, we end up forming 2 ATP. If you're keeping track and counting, that means we're at a zero loss of ATP at this point. So we're kind of even Stevens now. All right, we cost us 2 ATP earlier. We've just got 2 back. Even Stevens, we formed these 2 NADH molecules. Not exactly sure what they're about yet. We'll talk about those later. As glycolysis continues, we do form 2 molecules of water. Not super essential for you to know, but I will ask you that at some point. So just remember that, yeah, we do form water during glycolysis, 2 molecules. The 10th reaction, we form 2 more molecules of ATP. Again, we're taking ADP from the muscle. Hey, if glycolysis is active, you know the muscles are splitting ATP for energy, so you know there's going to be ADP around that needs to be reformed into ATP. So glycolysis, thankfully, is able to do it again right here at the last step. So we end up with 2 more ATP. Finally, after that stage, we end up with a molecule of pyruvate. Pyruvate is considered an end product of glycolysis. Maybe not the end product of glycolysis, but is a end product of glycolysis. So we end up with pyruvate. Now the story can continue and will continue with pyruvate making its way over the mitochondria. However, pyruvate can also continue down a different path, and it can be converted, oxidized into lactate. And so it has 2 fates. We'll talk about those in detail as well. But at the very least, understand that, yeah, that's the end of glycolysis. And we'll say pyruvate is going to be the end product for our journey, at least for today. So let's summarize glycolysis. We start off with glucose or glycogen. They're converted to glucose 6-phosphate. That's broken down by 9 more reactions that form basically glycolysis, right? The details of each of those reactions not critical for this class. The important products of glycolysis, the 2 end products, yeah, 2 molecules of pyruvate. We end up with a +2 on ATP, and that's of course assuming we start off with a glucose molecule. If we start off with a glycogen molecule, we end up with +4. We end up with 2 molecules of NADH. No, we end up with +3. Sorry, not good at mental math. 2 molecules of NADH and 2 molecules of H2O. So that's sort of the big picture summary kind of stuff. As I mentioned before, pyruvate can be reduced to lactate in the cytosol, right? So in the aqueous environment of the muscle, or the pyruvate can be transferred into the mitochondria and then go down further steps of metabolism, which is what we're going to be talking about next. Although I do want to talk about lactate for just a second. First, let's look at the conversion of pyruvate or pyruvic acid. They're kind of one and the same. Conversion of pyruvic acid to lactic acid. The lactate dehydrogenase reaction converts pyruvic acid into what's called lactic acid. Lactic acid, when it's formed in our body, immediately loses a hydrogen and becomes what's called lactate. So in that process, in that lactate dehydrogenase reaction, it actually takes 2 hydrogens to complete it. And then ultimately, yes, we end up with lactic acid. Lactic acid, for the sake of argument, and when people ask, yeah, it's not really present in our body for any meaningful amount of time. Lactate, however, is, and we can hunt lactate down and find it quite simply. Some of you may have done lactate testing in the laboratory before. So let's talk a little bit about lactate. So firstly, yeah, lactate production can result in the decrease of pH unless the hydrogen ions are buffered. And so our body has buffering mechanisms to help deal with changes in pH. Here's something worth noting about lactate. Lactate's been sort of vilified in the world of exercise physiology for quite a while, probably the last decade or so. A lot of the work done by Dr. George Brooks over at Cal Berkeley, but also other exercise physiologists around the world, have kind of started to better understand what lactate's role is in metabolism. And I think it's been, or it has been, based on all the research evidence that's starting to mount, very misrepresented. So firstly, this quote, each pyruvate molecule absorbs two protons into its structure, converting to lactate. Thus, lactate production is actually a consequence of cellular acidosis and not the cause of acidosis. So it's because of the presence or the release of those ions that lactate is formed. So the formation of lactate doesn't cause the acidosis, it's the acidosis that causes the formation of lactate. And that's at least a good frame of mind to adapt or to adopt. Increasing exercise intensity results in increased lactate production. So the higher exercise intensity, the more we're going to rely on glycolysis. Therefore, the more pyruvate's formed. Therefore, the more pyruvate gets converted to lactate. Interesting things about lactate. One thing is that lactate can actually be metabolized and used for ATP production. It gets converted back to pyruvate and enters the mitochondria and basically goes through the Krebs cycle and electron transport chain, giving us ATP. Some of the research as to why trained athletes have lower lactates for a given exercise intensity compared to untrained is partially thought to be because they're able to utilize that lactate as a fuel source much more efficiently. So it's basically disappearing, more so than untrained athletes who don't quite have that same capacity. So lactate being sort of vilified over the years. There's other things about lactate that are important. This isn't an exercise physiology class, so I don't want to get on too many tangents. All right, so let's talk a little bit about the NADH the NAD and NADH that we identified as being sort of products formed during glycolysis. And NAD, and there's actually another one called FAD, which is flavin adenine dinucleotide, are considered to be hydrogen carriers. And so I use a little taxi graphic on the bottom there. So just think, they're like a taxi cab driving hydrogen around. So NAD and FAD, they can accept protons. They can accept hydrogens and shuttle them to places where they need to go. And so when they're reduced, they are carrying those hydrogens, and they play a really important role in metabolism because hydrogen becomes an important player in our ability to generate ATP, especially in the electron transport chain. And so I will refer to NAD and FAD as the taxi cabs. So that NADH, right, from glycolysis, we didn't have FAD in glycolysis. So NADH from glycolysis, well, how does it get to the mitochondria, right? Because the glycolysis is happening, and this is something we didn't talk about before. We knew all this was going on in the muscle itself. Glycolysis is going on in the cytosol. It's going on in the aqueous environment, right? It's going on in the fluid around the muscle fiber. It's not the fluid in the muscle fiber, but it's not going on inside the mitochondria. And so how does the NADH get to the mitochondria? Well, we need another carrier. So we have these taxis that the hydrogens are being carried around in, NADH being a taxi, and it needs to be shuttled, and it is shuttled. And there's two different shuttles that take NADH to the mitochondria and drop off its passengers that were once in the taxi. They were not in the shuttle. And so these two shuttles, one of them is called the glycerol phosphate shuttle. And this exists primarily in skeletal muscle. And what it does is takes that cytosolic NADH, the NADH formed from glycolysis, and it brings it to the mitochondrial membrane. And in the process of going through the membrane, the NADH becomes FADH. So something happened to the passengers that were once in the taxi. I don't know, maybe they changed clothes. Something happened, now they're a little bit different. The NADH from glycolysis became FADH in the mitochondria. Not to worry, we still get ATP from the FADH, not as much as we do from NADH, but we still get it. The other shuttle is the malate aspartate shuttle, and this exists in the heart, the kidney, and liver, and it takes the cytosolic NADH, and when it gets to the mitochondria, it actually keeps the passengers intact, and they end up in the mitochondria as NADH. So there's really no change in the conformation of those protons that are being shuttled around. Hopefully that makes sense. So two different ways that the NADH goes from the cytosol to the mitochondria. Glycerol phosphate, malate aspartate. In the skeletal muscle, it's mostly glycerol phosphate. All right, So we finished stage one. We've talked about glycolysis. We start off with the glucose molecule, maybe glycogen. Glucose went through that whole process of glycolysis, ended up as a pyruvate molecule. How many carbons are in the pyruvate? Pyruvate's a 3-carbon structure still, so we still have these 3-carbon structures going on. Now we're going to talk about stage 2, which is the conversion of pyruvate to acetyl-CoA. So the formation of acetyl-CoA begins with that pyruvate. That's actually 2 for each glucose molecule. Starts with the pyruvate from stage 1, from glycolysis. It doesn't produce any ATP. No ATP produced. But we do get 2 NADHs, which is good. This occurs in the mitochondrial matrix. So this is going on in the mitochondria. It still doesn't require oxygen, right? We haven't talked about oxygen yet. But what's interesting about this stage is that if oxygen isn't present in the cell, it won't happen. If oxygen isn't present in the mitochondria, the shuttling of NADH to the mitochondria will not occur. Pyruvate will not go to the mitochondria. Pyruvate will become lactate. So things really slow down metabolically if there's no oxygen around. Pyruvate dehydrogenase is part of the complex that is responsible for this transformation. And the shuttling of pyruvate is done via a protein carrier. We don't talk too much about that. All right, so stage 2. Here it is, quick and dirty, quick and simple. So pyruvate, which is up here, is a 3-carbon structure, is converted to something called acetic acid. And so pyruvate to acetic acid, pyruvate will lose a carbon in the process and actually will form carbon dioxide. So now acetic acid is no longer a 3-carbon structure. We have a 2-carbon structure, acetic acid. Acetic acid, and so yeah, that carbon dioxide that's produced, it diffuses into our bloodstream and we exhale it. We just dispose of it. We don't need it. It's no good. Acetic acid that's now formed combines with a molecule of coenzyme A. Acetic acid plus coenzyme A gives us a molecule of acetyl-CoA, or acetyl-coenzyme A, but it's usually termed acetyl-CoA, just the abbreviation. In that process, an NAD then forms an NADH. So we have the formation of an NADH. Because we started with 2 pyruvate molecules, we form 2 NADHs and we form 2 acetyl-CoAs in this process. We also form 2 carbon dioxide. Pretty straightforward process. It's really important though. Acetyl-CoA is literally like a magical substrate. It is the universal substrate of metabolism. Carbohydrates, fats, and proteins can all become acetyl-CoA at some point. Your brain will actually live off acetyl-CoA. When it combines with another acetyl-CoA, it becomes a keto acid or ketone. We'll talk about ketones quite a bit throughout this term, but ketones are a pretty fantastic fuel source. So acetyl-CoA, the formation of acetyl-CoA is a pretty big deal. All right, that is it for this lecture, or for this part of the lecture. We will continue with part 2 next time.